please.
Thank you. Okay, well, first of all, that dong that we get before it is the best sounding dong I've ever heard in my career. I also want to thank everybody for coming to my talk on Sunday morning. And in terms of knowing everything, actually, as I tell my children and my students, the more we're in this, the less I know. The difference is I know that I don't know. That's very important. And by the way, I'm from Texas, which may be part of the United States, but that's debatable. Okay, so today what I want to talk about is what I call truly engineered approaches. And this, I don't know if Dr. Del Nito's in the audience, but Dr. Del Nito and I have had this running joke for a number of years, and it really comes from him, and it's that the whole purpose of all of this work is to turn surgery from a religion to a science. That I don't know, I certainly don't claim to be able to do that, but I think we're trying to make some baby steps in that direction. So based on my talk yesterday, I've removed every equation from this presentation, but if you want to see them, I got them. Okay, so I'm going to be talking about mitral valve disease mostly, and of course all of us, you guys here know far more about this than I do, and one of the things that we're trying to do is understand how to improve the use of various surgical modification techniques such as annuloplasty ring and so on. Really what appears to be a very, at least from my end, from an engineering point of view, a relatively straightforward thing to do, but it's actually extremely complex. Now, I want to make another disclaimer. I am by far and above not the first person to model the mitral valve. Some people in this room have done phenomenal work in this area. And actually, I was a classmate of Karen Kunzelman. We did our PhDs together at UT Southwestern Medical Center in Dallas back in the early 90s. And I believe she was one of the first ones to actually develop a finite element model of the mitral valve. And there's been many since. And there's also been some beautiful work by Dan Einstein, who works with Karen Kunzelman on fluid structure interactions in the valve. Some work, one of my former students, I think from Dr. Radelli's group, and so on. Lots of great people doing a lot of really great stuff. But you have to understand that this is not a bridge or it's not a simple structure. These are extraordinarily complex structures. We did these micrographs a couple years ago on the different layers of the mitral valve. And the first thing you notice is that, it's multiple layered, and we know this, but the question is why? I mean, we talked a little bit about that yesterday, and it doesn't make any sense to me. Why would it be multiple layered? It's a simple flap, right? But it also, in addition to the tremendous structural differences, I want to also draw your attention to the fibrosa, which is the major load-bearing layer, and it's made of a phenomenal, highly dense collagen. Those of you who have ever been to the San Francisco, been to the Golden Gate Bridge, there's a little park by the bridge, and they have a cable, a section of a cable they use to make the bridge, and this thing's about at least this big around, about 1 meter in diameter, and you can see the thousands and thousands of little cables they use to build it, and I always think of those things when I see these types of micrographs here. And you can see that. And of course, buried in those are the interstitial cells, as I talked a little bit about yesterday. So they're sitting within this extremely dynamic, dense connective tissue environment, and they play a really important role. So the question is how can we link these organ level models, so what's going down, way, way down at the cell level, which is ultimately driving all of this remodeling anyway. And of course, as I mentioned yesterday, these interstitial cells are extraordinarily complex cells. I don't claim to understand everything about these cells. There's been some beautiful work on the family of myofibroblasts over the years, particularly a guy named Boris Heinz, H-I-N-Z, has done some phenomenal work on characterizing these guys. A very strange cell, very phenotypically dynamic, has fibroblast and smooth muscle cell characteristics and so on, and actually it's responsible for maintaining and remodeling all of this. And as I mentioned to you yesterday, it does this through an extremely complex regulated processes where you go from quiescent to activated, where it's excreting collagen and elastin, the MMPs and TIMPs and all this, and GAGs and all this stuff being dynamically going. So one of the things we're asking ourselves is how do all this low-level cell level work gets regulated all the way up? And the other thing I want to underscore is how tightly they're coupled. Those are collagen, that's elastin, there's the cell membrane, and everything's tightly bound and in there. And so there's a lot of, the communication is largely physical and mechanical. There's actually direct connections there. So, as I mentioned to you, I like to use the term multiscale, although those of us in engineering know this term is really abused, and it's sort of like a full-service bank. What does that mean exactly? And the key idea here is that, at the surgical level, you're mainly dealing with this and with the valve, but underneath it all, you have the tissues down to the constituent layers and fibers and down to the cells. And by this is a nice little micrograph, gives you an idea of the density of the cell network. All these work together and they all communicate and they're all part of a single system. Okay, so it's not like you can spend your whole life understanding any one of these in particular. Understand how they talk to each other is a whole different ballgame. So in terms of, now, how does this respond to like major questions? And one of the ones are the surgery-induced changes, induce local tissue stress. We think that affects tissue homeostasis as well as long-term repair outcomes. This is a hypothesis. Our goal is trying to quantitatively put this on some type of scientific underpinning. Now, again, the basic framework is that we have some type of surgical repair. This leads to excessive or, as I'll show you later, ultra-low or hypostress levels, cellular response and tissue remodeling, and ultimately repair failure. And this makes an extremely complex process look very simple. But the idea is to try to understand something about the mechanobiology in these overload or underload situations. And I'll show you these systems seems to have a sweet spot where it likes to want to keep itself in. So one of the first things we did is I want to expand a little bit of what I presented yesterday, mainly to work with one of my very nice doctoral students, Salma Ayub, and Chung-Hao Lee, who's now at the University of Oklahoma, on linking these. And as I mentioned to you, we did some very basic studies, and we'll take tissue from ovine mitral valves, porcine mitral valves, cut them into little strips, put them on these little spring mounts, and put them on a little bioreactor. We basically just cycled them to different stretches. Now, this may sound like a very simple thing to do, but it does a lot of interesting things. This color maps are the collagen structure, that is how the fibers align, and cooler colors represent more random. As you increase the strain, as you would expect, the fibers become more aligned. The collagen is undulated, it straightens out, fibers rotate, and you'll take a look, and there's a big jump in this at 30% strain. We think physiologically it's sitting somewhere between 10 and 20. So 30 really represents beyond, 0 of course is below. Now if you look at what the cells are doing, they of course get strained and they get deformed. And again, there's this huge jump at 30%, but there's a steady increase. And here one thing you notice is that the nucleus and the cell membrane will really deform more at the very high strain levels, where here this is a little bit more normal. Now, what happens after you do this for 48 hours? And what you find, these open white bars represent the control, that is what the tissue was before it started, and the black represent after 48 hours. And what you notice at 10% and at 20%, there's not much change. At 30% strain, if you can really see this, there's an enormous upregulation. This is about 20-25, and these guys almost double. And all we're doing is changing the strain level on the tissue. There's nothing else that we're inducing. And so we can induce these enormous increases in soluble collagen output 48 hours simply by changing how we deform the tissue. That is how we deform the cells. Now, the other interesting thing is you look at the static, it goes down. So the cells need to have a physiological deformation constantly going in order for them to maintain their outputs. Whereas here, if you don't do it enough, it goes down. If you do too much, it goes up. So it's a little bit like Goldilocks and the 3 strain levels. It goes down and goes up. Sulfonated gags, you see similar behaviors. And elastin, very interestingly, elastin, at least we have evidence for. I think this is still, I'm not sure if I completely believe this, but there's evidence that elastin gets downregulated, possibly disrupted or enzymatically broken down. That may be very possible. Okay, so just what it seems to be really simple experiments, just beginning to understand how dynamic these systems are. Now, one of the things we also looked at is the layer-specific differences, and if you're very careful, you can peel the layers apart to some extent, okay, and you can actually break them up, and they split kind of along the spongiosa area. And what you find is that the fibrosa is what really is most of this. Atrialis gets also upregulated. Ventricularis pretty much just sits there and looks at you. It doesn't really do a lot. So already we're seeing that not only is something as simple as strain having this, different layers will respond, the cells in different layers respond differently. And this is largely because, as I'll show later, they sit in very different microarchitectural environments. And then not too much. Now, when we look at the genetic upregulation, what we've seen is that at 20% strain, there's some upregulation. I don't know if you can see this. The little red dots indicate upregulated genes. At 30% strain, you see a great deal more. So what's happening here is that underneath this is that cell deformation is triggering the cells to become more synthetic by some level of genetic upregulation. And again, it's all within 4 to 8 hours. It doesn't take a long time. Okay, that's all kind of interesting, cool. What does this have to do with a working valve? And this turns out to be not that easy to do, because one of the big challenges in biomechanics is you can take a tissue or some biological structure out of the body, you do some tests on it, but you really have no idea how to put it back in. And this has actually been a huge problem in the field. And we did this by going back and mapping the cell deformation. So we did this using little pieces of leaflet about a centimeter square, and we put them on a little biaxial stretcher, and it's under a microscope. We can image the cells and the connective tissue simultaneously, and we load them to the best of we can tell, approximate their physiological loading conditions. And then here's an example of the transmural structure of the cells, and you just go through this image processing process to extract these out. And then what you find out is that as you stress the tissue, the cell's nuclear aspect ratio goes up, but the fibrosa ventricularis go up a lot, atrialis mungiosa not as much. Again, there's that layer-specific differences. And then we developed what's called a very simple multiscale model. This was published in the Journal of Theoretical Biology about a year ago. Where we actually can do a model that this represents the approximate cell orientation and density, and then we put that into the model and we say, can we simulate what the cells are doing within that? And then we actually use, I don't know if you can see this, these are sonocrystal data. And this was a really fun little study I did with Rob and Joe Gorman, who I've been working with for many years. They're wonderful to work with. I'd call them up on the phone and say, I had this crazy idea, why don't we try this? They go, okay, sure. And about 2 weeks later, they send me an Excel file with the data. It's wonderful for me. And what we did for the first time was really measure the deformation of the leaflets. This is how some very basic information in literature just doesn't exist. And then we can actually simulate a little cube of tissue, and this is what the model does. So that little green block represents some arbitrary block of tissue inside the valve as it's stretching. The amount we strain it is either what we measured in vitro or what we measured in vivo. We can just arbitrarily assign that. And the little ellipsoidal blobs there you see are what we estimate how the cells are being deformed as the tissue is straining. Now again, I need to underscore that in vivo these are non-measurable quantities and you have to use a computational approach to estimate them. So you can see, by the way, that packing density of the cells and the orientation is taken from histomorphometric measurements, so it's not arbitrary. And as you can see, they move quite a bit. So what we end up doing is matching the properties of the cells to what we've measured in vivo. And we've done this a variety of ways. And again, if you're interested, I can send you the paper if you want the technical details. But the cool thing is that when we look at the different layers, is that the stiffness of each cell is about 5 kilopascals, all about the same. The differences in how they're deformed is strictly due to the microstructure in each of the layers. And that suggests that if the stiffnesses are the same, they may be phenotypically in similar states, but the synthetic levels are driven differently because they deform differently because of the microstructural difference. Again, all this is fine-tuning within each of the 4 layers. Now, if you want to go back and say, okay, we can take all of this now, and how do we put this into a functioning valve? Well, we go back to our sonocrystal data. We take the pressure, and we take the strains, and you've noticed the strains in the valve are quite large. And you notice the valve also loads very rapidly in about 1/10 of a second. So when that little animation I showed you, that stretching occurs in the snap of a finger. It's very fast. And then from the model, here's the stretch we measured in vivo. And here's what we estimate how the cells are deforming in each of the 4 layers, noticing that the fibrosa is the highest. and it loads very rapidly, and it's held and goes back down. So this actually tells us kind of where we are. And then the other thing is what happens if you put a ring on it. And if you do that, the ring goes here, and what the ring does is to lower the circumferential strain, at least in that small region where we've measured it. So this is what the repaired valve works, and it actually lowers the total strain of the cell. And so to summarize, this kind of wraps it up. So this says that if I put a normal valve and I measure how it's loading, the cell will go up to about an aspect ratio from about 2.5 to about 5, so about doubles, okay, meaning that it elongates about like that. Now, if you put a ring on it, it means that in that central portion, and again, I'm not talking about what goes on in the rest of the leaflet, just in that zone that we can measurement, goes quite a bit less, about 3.28. So one of the things that a ring might do is not to overload, but in some cases underload the tissue. And based on what I've showed you before, we came up with this sort of little graph where we say if the cell deformation is within this roughly 3.5 to 5 zone, the tissue will stay in more or less homeostatic conditions. If you overload it, it goes into hyperphysiologic mode. But interesting, if you underload it, which is what we think was happening here, so that's 3.28, this is right about there, you actually will start getting downregulation. It could eventually lead to a breakdown or creep of the tissue. Again, this all has to be proved. This is all speculative on my point, but at least we have some indication of doing that. So just to summarize, we found that the big reduction in the aspect ratio when you put a ring on it, nonlinear increase in tissue micromechanics and reduced stresses, direct dependency on tissue stress, and the differences between hyperphysiological loading and hypophysiological loading. So the main point here is that you can not only overload, the natural inclination to think that it's overloading the tissue, but it's probably the other, or at least as likely. So one of the questions is, this is all very nice and it's great science and all that, but do we have any evidence, what do we really know how mitral valve or valvular tissues remodel at all? And one of my former doctoral postdocs, Sarah Wells, was associate professor at Dalhousie University out in Halifax, did this really grisly study where she went to the slaughterhouse and actually got bovine mitral valves from pregnant heifers and did a very detailed analysis of it. And she's looking for basically, does a mitral valve remodel under pregnancy? And I think most people know that it does in some case. So what pregnancy does, in fact, we have this running joke that pregnancy is a disease. But anyway, if you take a look, what pregnancy induces is a volume overload model in an otherwise completely healthy system. It displaces the papillary muscles and causes a substantial amount of annular dilation. So essentially, this can happen in infarction, but in the pregnant case, what you're doing is stretching out the leaflet and the papillary muscles are pulling down. And the leaflets, by the way, are not pathologic, and they must grow and remodel to adapt to these altered boundary conditions. So another way to think of pregnancy is that pregnancy induces altered boundary conditions. We also thought that was kind of amusing. So what do we know? It says we know that in terms of cardioeffects effects of pregnancy, it increases blood volume, cardiac output, LV mass, stroke, 30% increase in stroke volume, and 12% annular dilation or orifice area. And the question is that we go from conception to early pregnancy to mid-pregnancy term. What's happening here? So I know obviously this is not meant to be read, but Sarah did this beautiful work with very detailed mapping out the amounts of collagen, wet weights, dimensions, thicknesses, layers, the whole, mechanical properties, the whole bit. And it turns out there's actually very substantial changes in the leaflet. Now, if you take a look at, I'm going to score some of those, just look at surface dimensions. If you look at the circumferential distance, that's just you cut the anterior leaflet out, measure just its width. It will start off here at about 4.5 centimeters and gradually increases to term to about 6. The radial is also increases, but not as much. And if you look at the linearized rates here, you actually see they increase about a little over 1% per week in the circumferential and about a little over 0.5% per week in the radial. So what you're seeing here is not only gradual increases, but directional differences. So it's not an isotropic dilation. And if you look at the mechanical properties, if you take a little square piece of the tissue and stretch it in 2 directions, the so-called biaxial test, you'll see this. And what happens here is really interesting. It starts off with non-pregnant, in early pregnant it becomes stiffer, and then by late pregnancy it shifts back. So there's evidence of some type of tissue remodeling or adaptation process there, and that's what got me very excited about this. So we said, how are we going to model this? How are we going to put this in and try to get something out of it? And we started with our so-called structural models, and I just kind of overview this just for general interest. And we really start with the level of the fiber. If you go all the way down to the molecular level, the collagen that makes up most of these valves is a tropocollagen. Now, those of you who are familiar with DNA, that's double helix. This is a triple helical structure, and it's really designed to be like a rope. You have alternating left-right handedness, and the whole structure of collagen is a phenomenally efficient system, and you go all the way up to the fibril to the fiber. But we're starting really here just at the fiber. We're not going to concern ourselves with what goes down underneath here. This is already hard enough. Now, this is a really nice three-dimensional reconstruction of some mitral valve collagen from the fibrosa. Now, note some interesting things. It's undulated, they're densely packed, but there's also a fair amount of spaces between those fibers. Okay, and the question is, how do we model that? Well, we just look at a single fiber, which is undulated. Okay, I'll go with the direction. Now, in terms of the model, there's actually a lot of evidence in the literature that indicate that the collagen is a linear, just like a little spring, but it's slack. And so what that means is that as you take some strain before it'll bear load. Okay, but remember that means it's a fiber is linear. Now, we will then look at what's called, start integrating this up the scales. We'll say now we combine all the fibers that have a common orientation. We call that an ensemble. And then what it is that even though the fibers are linear, as more recruit, in other words, as you pull it, then the ones that have the smallest undulation stretch first, and then gradually all get acquired to the full, and that gets you this nonlinear stress-strain curve. And you actually come up with statistical distribution for the undulation. Again, I can explain details of this later, but it's essentially the way nature fine-tunes that stress-strain properties with linear fibers. And then we can also integrate over multiple angles to get all the fibers and all orientations. And then what you end up getting all of this is that you can back out from that stress-strain data how much the elastin contributes and how much the collagen. So by the way, most people would say the elastin doesn't do much, but the elastin is actually really important because we have new evidence indicate that in vivo, most of this stretch that you see here is occupied by the pre-strain of the leaflet, and that the elastin is keeping the leaflet very taut. But clearly the collagen is playing most of the mechanical role. So what happens when we look at this, if we look at how the fibers are oriented in the leaflet, you go from non-pregnant to to late pregnant, and then back up. So there's this sort of, actually I take that back. It goes from non-pregnant to early pregnancy, and then it stays this. So that means the fibers start more aligned, and then they become less aligned with pregnancy. And you can see that with the standard deviation here. So imagine these are Gaussians, these are standard deviation. This is the non-pregnant. It goes down, and then it gradually increases all the way up, and it keeps going up until term. Now, the other thing, and again, this may be a little subtle, but what we're finding is that the fibers, as they stretch out, they actually get stretched out, and then over time, the tissue remodels to recover that recruitment. That is, you go from non-pregnant to fibers being straightened out, and then back to late pregnancy. And you can see that, and I'll show you a little bit more. Now, the really cool thing is that Sarah didn't do, we went back and got her paraffin blocks, and went back and said, what are the cells doing? Guess what? It's not random. This is what the cells look like. Notice that highly undulated collagen fibers. Now see how much straighter they are? That's what this shift is telling you. It's saying the fibers start being undulated, they get straighter, and then they go back to being more undulated again. This can be, if you look at the nuclear aspect ratio, it goes up and then goes back down, and we've actually modeled this, but what's interesting about this is that what's happening here is it seems that the cells are remodeling the collagen to get themselves back to the shape they want to be in, and they do this by making the collagen more undulated. And I think this little graphic here kind of shows overall what I've been talking about. So you get this initial undulation, and you strain it, and then during, say, midway to third through pregnancy, things get strained out because the leaflet's being stretched out. And then for the rest of the term, which is about 100 days, it's actually adding material in series, to the existing collagen fibers, as well as adding new material so they can go back to their original shape. And this, I believe, is the first evidence of its kind in valvular tissues of this kind of remodeling process. So these are the kinds of things we also think are happening in things like repaired valves, but again, there's no evidence for that yet. Okay, you get this picture. How do you use this practically? And we haven't done that yet, but we do this by developing these very detailed mechanical models of the mitral valve. And this is from one of my former postdocs who actually put together. And it turns out doing all of this work is a great deal of work. And there's all these multiple paths. You have to get the geometry, you have to get the tissue behavior, you get the morphology. A lot of the organ geometry was done with my colleague Ajay Yoganathan, a very well-known investigator in heart valve fluid dynamics at Georgia Tech. We can then actually do micro-CT reconstruction. Again, this is not really new. Lots of people do this. And then we can actually markers together. The thing that we can do is we can take those collagen maps that we measure, and then we can actually map them onto the structure of the leaflet itself. There's an example of ovine mitral valve. Now, what's really cool about this is that what this tells you is that there's more going on than just stress. Okay, so we tend to think about things somewhat practically. What's the maximum stress or strain? But what I'm plotting here isn't stress, it's the collagen alignment. And you can see this in the animation. The valve closes, you see, as soon as those leaflets coap, that collagen alignment goes way up using this color scale. And you can see that here incrementally here. Okay, and then we said, okay, we can do this. What if we change the properties of the collagen? What if we make the collagen fibers very linear or much more curved? Does it really matter? And the answer is what we find here is that it's very interesting. This represents the normal valve. This is the total stress. See the hot spots in the center. This is the degree of orientation. If I make the fibers very linear, okay, then the valve becomes very you can see a lower degree of stress, but you have a much higher degree of recruitment. So all that structural reserve, the mitral valve, is lost. If I make the fibers too curved, the stresses go up, but there's not much change. So that system is really designed to make it optimal. So how is that useful? And you can kind of see that here. Sort of normal is here. Overcurved here and straight over here. So you can see the lowest tissue stress may not be a single indicator for a functioning valve. Collagen fiber architecture and structural reserve are critical for maintaining proper function of the valve, and the balance between tissue stress and fiber architecture could provide guidance in designing optimal surgical repairs. And I'll get into that in a little bit to show why we think there's some evidence for that. So one of the problems in developing these models, just to kind of wrap up, is that clinically, while you can get the leaflets, you can't get the chordae. So what do you do about that? You really can't get patient-specific chordae. And we're completing a study now where we looked at the ovine chordae here. And what you see here is the collagen network, the chordae network. And we actually went back to our 3D reconstructions, segmented the branching structure all the way down here, and then began to look at the different chordal insertions and actually begin to get maps of these things and how do they work. And it turns out that they follow a very nice branching structure. These little guys here are called Reeb graphs, and you can actually take them and structure them together. And they found these very interesting patterns. And in fact, you can see that for we've actually done 10 valves now, which got the paper all done. I warn you, don't do this in your spare time. It took us 2 years to get all this done. It's unbelievable. It's easy. Just get the chords, you'll just digitize them, and it's just, you don't want to know. But what's cool about it is that there are consistent patterns. Now, this is in the sheep. We don't know what goes on in the human, but it does suggest that you might be able to get population averages, that you can construct population-based chordal structures. As far as I know, nobody's ever actually looked at this before, and now we know why. Okay, now again, we're looking at, in our collaborations with the Gorman Lab, they're very keen on, they've been looking at valve shape over the years, and we're trying to figure out we're trying to say how we can adapt our models to fit these. And one of the things that you can do now is to say, can you use all this information we've collected to design a better AP ring? For example, the idea is not just coaptation, not just stress, those are important metrics for sure, but try to get the collagen structure and ultimately the cell deformations in an optimal sense. And maybe, one of the things we find, by the way, in the mitral valve, is that you can use a number of different material models and you end up getting about the same simulation accuracy in terms of the shape of the valve. But boy, this internal structure and stress system really change just by subtle changes on the annular geometry. Now, again, this is a big step in terms of proving this in an animal, ultimately what you'll see clinically, but it does suggest there's at least some wiggle room in terms of using all this information we've learned to improve a very basic therapy. And I wanted again to say that if anything I can take credit for, is just getting really great people to work with me, particularly Chung Hao did the modeling, Bruno Rigo who did all the analysis in the pregnancy, Sama did all the cell level, Amir who's doing all the chordae, Zhao and Yusuke Sakamoto help with all the modeling work. And of course the generous funding from NIH and also really great collaborators, Joe and Rob Gorman, who are twins, by the way, and after all these years, I can finally tell them apart. So that's want to thank you very much, and hope that was educational.